All right, so there's only a couple slides that I wanted to cover from wrapping up the muscle thing, and it's only because I don't remember who it was, but someone over here, one of you two, had asked about it, someone in there. There are three types of muscle fibers. There's type 1 and type 2A and B, or sometimes called X. So here this is depicting two. Fast glycolytic and slow oxidative. And this is a cross-section of some muscle, and the light colored tissue is fast glycolytic. The dark is slow oxidative. These slow oxidative muscle fibers look like that because they're highly enriched in myoglobin. And I had already said that before. I had talked about myoglobin being a single subunit that is related to the tetramer, which is hemoglobin. Okay, has this porphyrin ring which contains iron and binds oxygen, and so this serves as an oxygen reserve in the cell. All right, because of this heme group, this porphyrin ring with the oxygen in it, these slow oxidative fibers are stained darker. They look darker red. Okay, and this, in fact, is the difference between, in a chicken, white meat and dark meat. Okay, some fiber is not meant for long endurance, and yeah, Jacob.
Oh, just something kind of cool that in comparative vertebrate anatomy, some frogs and fish actually have fast oxidative fibers as well.
Yeah. So, human anatomy here, though, comparative anatomy is interesting. The different strategies that organisms come up with. Humans have three different fiber types. And you've seen this diagram already, but here I have it tied to some muscle fiber types, so you can begin to see that an eye muscle has significantly more fast glycolytic fibers, and a gastrocnemius is going to have more of these slow oxidative fibers in the mix, and then the soleus is going to have a considerably higher ratio of the type 1 fibers. All right. So these fast glycolytic ones are not fatigue resistant. They wear out quite quickly. They have lots of enzymes required for the phosphagen system and the glycogen lactic acid system, where we're breaking down glycogen, throwing it into glycolysis to produce pyruvate, which then gets converted to lactate and exported to the liver for the Cori cycle, if you remember all those steps that we talked about. Do not have a lot of mitochondria, right? Because they're fast glycolytic. They're not using oxygen. They don't as much oxygen. They don't need as many mitochondria, and there's not very much myoglobin. All right. So, and on the other hand, there's these slow oxidative fibers, which have lots of mitochondria, lots of myoglobin for oxygen reserves, and there's a lot of capillaries. So comparing these fiber types, we have type 1, slow oxidative, and the two fast glycolytic, the type 2A and 2B. Their cross-sectional diameter varies from small in the slow oxidative fibers to quite thick in the fast glycolytic type 2B. And the tension that those fibers are able to create varies across the fiber type. Of course, the speed of contraction varies amongst them. A slow oxidative fiber, as I've said, is very resistant to fatigue. Yes?
So are the three types those three, or are they fast, intermediate, slow? Is one intermediate?
So there's a slow oxidative type. There is an oxidative type that is a little bit faster. We call this a fast oxidative type 2A, and then there's a fast glycolytic type 2B. In the previous slide, I just showed that there were 2, but there's actually 3. There's a fast oxidative and fast glycolytic. He was referring in frogs to a fast oxidative type, and there is a type 2A in humans. So, and it's this fast oxidative type that does, in fact, have a lot of myoglobin as well, and an intermediate number of mitochondria. There is a high concentration of glycolytic enzymes. So this 2A is sort of a combination. It has some of the benefits of both types. However, it doesn't have the extremity of the benefits of either. So it truly is as kind of a synthesis of the two fiber types. All right, and then this is the last slide I was going to talk about. When you can change the expression patterns of different fiber types in your body by usage. All right, so here most of us in this room are probably, a lot of you are athletes, maybe not everyone is here, but so an average person is going to have about half of the fiber types are going to be the slow oxidative type, okay? And then about the other half are going to be fast types 2A and X, that's the same as 2B. Now, however, if you were compare an active individual with, for example, an extreme endurance athlete, now the muscle fibers that you're going to find in, and this is one of Andrea's slides, so I haven't seen the annotation on this slide. This is one of the only slides, maybe the first slide I've shown of hers all semester. So I don't know exactly what this means percent of total muscle. Perhaps that's throughout the body, although I have a hard time believing that. It seems like that may be total muscle in the calf. I'm not sure what that's referring to, but it's showing some reasonably comparative sample of muscle across the body. In an extreme endurance athlete, the percentage of slow type 1 is going up. And then a person with a spinal injury who is not, who's basically paraplegic, all of those slow oxidative fibers are dying back, and you're getting a preponderance of the slow or the fast glycolytic and the fast oxidative type 2 fibers. All right, and you can see the spectrum in between. Are there questions? You were sort of waving.
Oh, yeah. So, what's it called? When the various ratio shifts of fast to slow, that's just the diameter of the fibers, or is that also a production of new fibers?
That is the expression patterns of the fiber. So, I mean, you're turning over myofibrils, right? So they, with time, myofibrils will degrade, and you'll grow new ones. They'll be replaced or repaired by myosatellite cells, and so that is responsive to the demands put on them. I mean, we all are aware that if you go to the gym, you start pumping iron, you're going to be building muscle mass, and so that's going to be changing the composition of the muscles and the distribution depending upon the type of demand you're putting upon the muscle groups. Are you doing a lot of aerobic activity, or are you doing a lot of anaerobic activity, or are you not using muscle and you're losing muscle mass and thus decreasing this type 1 fiber?
I guess what I was asking is, do the fibers get thicker, or do you get more fibers?
Oh, I see. Both. So a fiber itself can put on mass, but you also are going to be generating new fibers. Yeah.
Okay, and that both mechanisms contribute to that shift, or is it only the generation of new fibers?
Yeah, you're not
preferentially increasing the size of one.
No, once it's a slow oxidative fiber, that's the fiber type that it is.
I didn't mean changing the type, but I meant like your slow oxidative fibers will increase in diameter faster than your fast twitch, or is it that you're making so?
It's dependent upon the demands you are placing upon the muscle, the whole muscle. Like what are you asking of the muscle? Are you engaging in aerobic activity or anaerobic activity? And that's going to influence the muscle fiber subpopulations in those muscles. Okay, any other questions? I don't want to get bogged down here too much because I'm not going to test you on this too much, I don't think. But oops, let's take that out. That's my dad. Let's see if I can find. He had Lou Gehrig's disease, which is a de-innervation of. Where am I? This is it. Oh, here we go. De-innervation of the muscles and their death. Okay. Lecture 5. Topic 5. The nervous system. Nerves tingle and tell us of what we are, but not. No. Nerves tingle and tell us of what we are not, but what are we but nerves? All right. The word brain is has Proto-Indo-European origins, which is fun for me because most of these words that we use come from Latin or Greek, and so it's kind of fun to have a word that is so central to the human experience that goes much farther back than that. It comes from mregmo, which means skull or brain, which then in the Proto-Germanic became bragnon or brain. Aristotle, we've talked in the first lecture I talked about Aristotle and how he believed that nerves were controlled by the heart. He was confusing them with ligaments, of course. And I've talked about Galen, who was pretty pleased with himself by giving the smackdown on Aristotle 200 years later with this quote, disparaging those who know nothing of what is to be seen in dissection. I have shown. Have I read this quote already? I feel like I might have. I have shown in my book on the teachings of Hippocrates and Plato that the source of the nerves, of all sensation and of voluntary motion is the encephalon, which is his word for brain, and that the source of the arteries and of the innate heat is the heart. A little plug for his New York Times bestseller book on the teachings of Hippocrates and Plato. The word nerve itself is indeed a Greek word reflecting Aristotle's misconception of them, meaning it is the word for sinew or tendon. A neuron means sinew or tendon. So we have retained that misnomer throughout the millennia since the Greek misunderstanding. I had mentioned Maimonides in that first lecture, I believe, as well. One who is not knowledgeable in anatomy may mistake ligaments, tendons, and cords for nerves, striking the same chord, as it were, as Galen 1,400 years earlier. Benedetti, about 5 years after 1492, right? 5 years after Columbus was setting sail, wrote, by means of nerves, the pathway of the senses are distributed like the roots and fibers of a tree. So here we're beginning to see an understanding that there is a connection between nerves and the collection of sensory information. That nerves are the conduits for information that is being gathered from the outside world and transmitted to the brain. So this is in the late 15th, early 16th century. Da Vinci, around the same time, said the medulla is the source of the nerves which give voluntary motion to the members. And yeah, he's not exactly correct in that, but he is still trying to draw a connection between efferent. So Benedetti here is talking about afferent, and we'll get into what these words mean, or sensory information. And Da Vinci is talking about efferent, or the control of the brain over the body. So Benedetti's talking about how the nervous system is collecting information, and Da Vinci is talking about how after processing that, the brain is able to control, respond to that. All right, so voluntary motion to the members. And here we have some very beautiful pictures, some diagrams that Da Vinci did of the nervous system at the time. So here is the most basic schematic of the function of the nervous system. We have the collection of sensory information by a host of different sensory organs that transmit that information up to the spinal cord, to the central nervous system, the spinal cord, which then, once it's in the central nervous system, either the spinal cord or the brain, goes through some sort of integration, integrative functions, where that sensory information is processed, and then there is the control. There is efferent output. There is control output that is sent to various muscles and organs and glands in the body. All right, so collecting information, processing it, and acting on it. That is the primary function of the nervous system. Here's a hierarchical depiction. There are three general categories of the nervous system, and these could be drawn with different connections than I have, but this is the way I've drawn it here. We have the central nervous system, which is separated into the brain and spinal cord, the peripheral nervous system, which has somatic nerves and autonomic nerves. The autonomic nervous system is divided into the parasympathetic, or rest and digest, and the sympathetic, the fight or flight portions. And then there is the enteric nervous system. This is a related but fairly autonomous set of neural tissue that guides the function of your digestive viscera. So the enteric nervous system is connected to the rest of this via the splanchnic nerves from the autonomic nervous system, but a lot of it is fairly independently functioning, which is why a paraplegic is still able to have intestinal motility. All right. Laid on top of this, there are three different ways, three different categories that are binary, three different binary categories for distinguishing neuronal fiber type. What kind of fiber is it? Is that neuron a general or a special fiber? Okay. Is that fiber somatic or visceral? This is in the peripheral nervous system. Is it somatic or visceral? And we're going to go through what each of these mean. Is it afferent or efferent? Right. Because of each of these three binary choices, it gives rise to 8 different combinations, or 2 to the 3rd power. All right, just like there are 6 sides of a cube, but there's 8 corners. It's analogous to that. There are 8 different fiber types, and these would be GSA, SSA, GVA, SVA, for example. So GSA would be a general somatic afferent fiber. All right, and SSA would be a special somatic afferent fiber. A GVA would be a general visceral afferent, etc. Okay, there are 8. This one, SSE, the special somatic efferent, is in shadow because there probably aren't really special somatic efferents. They probably are all general somatic efferents. Okay. Functional classes. This is getting back to that first slide that I showed of the very general schematic. I showed the collection of sensory information. We call these sensory neurons afferent neurons of the peripheral nervous system. They're sensory neurons, afferent neurons. So here's an example. The sensory neurons in your finger. They're collecting touch receptors. And within that, there are 3 categories. There are interoceptors, exteroceptors, and proprioceptors. Interoceptors are going to be sensory neurons that are monitoring the inside somewhere. You have a stomach ache, or your bladder is full, or what have you. You have pancreatitis, God forbid. That kind of thing is an interoceptor. An exteroceptor is, you can think about it as all of the sensory neurons that are pointed outward, that are giving you information about the external world. These are your external senses like touch, temperature, pressure, vibration, etc. And then any of the distance senses, so sight, smell, hearing. These ones here would be considered, for example, general senses, touch, temperature, pressure. Special senses would be like sight, smell, and hearing. Those would be special efferents. Yeah?
Now, is that just a functional classification, or do you know, like, I guess, embryologically, do these groups come from the same cell lines?
Okay, embryologically, different functional classes of neurons can have different embryological origins, for sure. But that level of detail is quite sophisticated. I do touch upon it at the end, on the 5th lecture here. I'll touch on that a little bit, but this is not an embryological distinction. Proprioceptors, it's a class, and I'll say this because each of these categories has multiple subclasses in them, which are going to have different embryological origins. Okay, proprioceptors are responsible for returning information about the position of the body, joints, and muscle contraction, state of muscle contractility. Interneurons, this is basically the central nervous system. These are neurons which are not gathering information or directly synapsing on an effector, all right, like a muscle or a gland. All right? So interneurons, these are the ones, interneurons are the vast majority of the cells in the nervous system. The vast majority is the interneuron, and we're going to talk about them the least because that is well beyond the scope of this class. But what you are, what you think you are, is a result of the function of these interneurons. And then motor neurons, efferent. So look, we have afferent is sensory, and efferent is motor output. And this is the same distinction between the word affect or affect and effect. So if you are affected by something, something is causing a change on you. And if you are causing an effect, you are causing that change on something else. You are it's heading out from you. So affect is heading towards you, and effect is heading away from you, as is afferent and efferent innervation. So motor neurons are efferent neurons, and the categories here, somatic and visceral. Somatic are just skeletal muscles. That's what a efferent somatic is. You're going to have skeletal muscles. And then visceral effectors are going to synapse. The visceral are going to be the autonomics, and we're going to have two types, preganglionic and postganglionic fibers. We'll talk about that when I talk about the autonomic nervous system. There's one lecture devoted to that. All right. So the central nervous system, its divisions. Very simply, there's the brain and there's the spinal cord. So there's going to be some sort of, whatever input is coming directly into the brain is coming along what are called cranial nerves. There are 12 of them. I am going to flash through them in a day or so here, the next lecture or two. And then the output directly from the brain is via the same cranial nerves. All right. So if sensory or motor output, sensory input or motor output directly into the brain are along cranial nerves. Then there's the spinal cord. Sensory information coming into the spinal cord comes along spinal nerves. All right? And there's reflex centers that happen in the spinal cord. They can synapse on interneurons where the signal is going to ascend the spinal cord. That is possible. But the original sensory information is getting in via spinal nerves to the spinal cord before it ascends. Spinal cord is central nervous system, right? All of this is central right here. But it doesn't have to ascend. If whatever sensory information is coming in is to reach your consciousness and for you to be aware of it, there will have to be an ascending fiber that synapses in the thalamus. But that is not required. You can have reflexes that are subconscious that then have motor output along the same spinal nerves. So it's cranial nerves going into the brain, spinal nerves going into the spine. And then these effectors can be muscle, smooth muscle, cardiac muscle, various glands, adipose tissue, etc. All right. Sensory receptors. There's 4 kinds of information that a sensory receptor can transmit. And we're going to go through each of these. The first type of information that a sensory receptor is going to give you is what is called the modality. The modality. And they say, oh, there's 5 senses, right? Well, that's actually a gross simplification of our sensorium. But there's taste, touch, hearing, smell, sight, right? These 4 here are each categories of special senses, and then this would be a general sense going back to those tissue categories, those fiber categories. So a sensory neuron is going to convey a type of information, a modality, a certain mode. And there's this notion of labeled line code. The information that is being sent back to the brain, it's just an electrical stimulus, right? But it has been mapped onto the brain. That neuron that's bringing sensory information of a certain modality into the brain is being mapped in such a way that it's interpreted by the brain as representing some quality in the external environment. All right? And we call this the labeled line code. The line that's coming in is labeled and thus interpreted in a specific way by the brain. Here's a taste bud. All right? And within this taste bud, there are different sensory neurons all clustered together. And this was a question about how taste worked for a long time. Even still, you can find textbooks. My in-laws bought a book about the body for my daughter for Christmas, because this is what she said. And I opened this brand new book about the human body for kids, and they totally got it wrong. They totally got taste wrong. They said that you taste sweet on the tip of your tongue and salty on the sides or whatever and sour in the back. That is not how taste works at all. You can taste all of the different modalities of taste on all of the places of the tongue that are sensory active. but there's this labeled line code. So if you taste something that is bitter on the central portion of your tongue, it is because you have stimulated these neurons, which are part of a labeled line that the brain interprets as being bitter. If you taste something salty in the center of your tongue, it's because you've stimulated these sensory neurons in that taste bud that are then interpreted via its labeled line, and so forth. There are 5, they've identified at least 5 different taste receptors. I've heard about the water receptor. I'm not prepared to talk about that. But bitter, so this is bitter, something that's bitter is alkaline, like an alkaloid, something that's basic, like baking powder, if you've ever stuck that in your mouth. That's going to taste bitter. Chewed up a pill, an aspirin or something. That's bitter, and it's alkaline, basic in pH. Salty, we all know. Sweet, of course, we know. Sour is acidic. It's the opposite of bitter. Sour and bitter are opposites, so like a lemon is sour. And then umami is the taste of glutamate. It's the taste of an amino acid. So this is telling us when things are savory. Umami is a Japanese word used to describe the sense of savoriness that is in food, and this is due to glutamate. Yeah, Jacob?
So then why are, I guess it's not a balanced pH, it's just if there's an acidic or a basic component in there, because like limes are both sour and bitter.
I'm not prepared to debate a lime, but yeah, perhaps you could be stimulating both of them. Maybe. I don't know. I'm not sure if limes are actually bitter or not. So the second type of information is the location. And we're going to talk about the sensory homunculus in the brain maybe tomorrow, Wednesday. There is a map of your sensorium in the brain. So there are specific brain regions that are directly hardwired to a specific spot on your body. And so a cell, a sensory neuron, is not only giving you the modality, but also the location of where that sensory information is coming from. This gives me a chance to introduce the concept of receptive field, the size of receptive field. So something that has a large receptive field means a single neuron is monitoring a large piece of turf. Okay? But since there's only one neuron, it's corresponding to a small region in the brain. All right? So because of this, if you take this 2-point discriminator, this is a lab that I do when I teach the lab in January, you have 2 points, and if both of those points are within the same receptive field, you will only feel 1 point. You'll only feel 1 point. All right? So places like on your back have large receptive fields because you don't need a whole bunch of high-resolution data from your back. You just need to know that, my God, there's a tarantula on me. Get off. Thank you. Don't need to know exactly where it is, just something's on my back. Whereas your fingers, your lips, your nipples, your genitalia, you need a lot of sensory information from those regions. You need a lot of fine point discrimination from your fingertips. So there's a very high density of neurons, very small receptive field. So the size of the receptive field is inversely proportional to your ability to locate that stimulus. All right? So here's the same area on your fingers, but now there's 3 neurons. So in the brain, there's going to be quite a bit larger area devoted to your fingertips. This is an important concept. We're going to hit it from another angle later. Third type of information is the notion of intensity. All right. So, and there's a number of ways that this kind of information can be sent. The intensity of a stimulus. It can be dependent upon which of the fibers. So again, this relates to labeled line code. if this neuron, for example, gets stimulated, the brain represents that as being of a different intensity than these other neurons, which are not firing. Maybe this is only very low stimulus is going to trigger this. So we say, oh, there's just a very mild stimulus here. All right. How many fibers are firing is also a way to encode intensity. So there's relatively more fibers here that are firing on the brain. And then the rapidity of these. Remember, I said that action potentials encode information by frequency modulation. Okay, how rapidly? What is the frequency of firing? All right, it's not the amplitude of the signal, because a signal from a single neuron is always the same intensity for that neuron. It is the rapidity. So here, this same neuron is firing twice as fast down here. So that's going to be interpreted in terms of a different intensity by the brain. Does that make sense? It's all pretty straightforward. And then finally, the fourth kind of information is the duration. The duration. So I'm finally cutting up the kind of information that your brain is gleaning from any sort of sensory input here. All right. How long is the signal lasting? How long is the signal lasting? This gives me an opportunity to introduce the concept of adaptation and two types of receptors, tonic and phasic. So adaptation is your body's ability, so I have it written, reduction in sensitivity of a constant stimulus. It's your brain's ability to filter information out. Often people think of the brain as this amazing organ for absorbing all of the information around it. This is not true. This is not true. Our brain is exceptionally good at not hearing things and picking patterns out which are meaningful, only absorbing things which are meaningful to us. You sit here, all of us in the room, I'm assuming that none of us are profoundly hearing impaired, and you are listening to my voice, which has, you could record my voice, do a Fourier transform on it, and look at the spectral window of my voice, right? You could look at the frequency density of my voice, the power spectrum of my voice, and that's going to have a certain spectral density function associated with it. That spectral density function is a characteristic that your nervous system has learned to identify in its environment as being meaningful, all right? And as such, worthy of your attention. The constant that we hear from the air handler or whatever that sound is, wherever it's coming, maybe it's the fan on this thing up here, it's here every lecture, every day, every moment, that thing's on. But you are not aware of it except when I point it out. Hopefully it's not driving any of you crazy. All right? Because it's not meaningful. The spectral density power the power function of that sound is not meaningful to you, and your body has learned to filter it out, adapt to that sound. All right? So the two fiber types I'm going to talk about here are tonic and phasic. Tonic are slow adapting receptors. They're always active. They do not have peripheral adaptation. All right. So, for example, proprioceptors. They're returning information on body position, muscle tension, joint motion. If I were to do some kind of yoga pose up here for you, I could sit there and hold it, and at no point would I forget that I'm holding it. Phasic or fast adapting receptors are typically inactive. They're going to give us rate of change of stimulus, the derivative of whatever that stimulus profile is. So examples are smell, When I was maybe the age of some of you guys, I didn't go to college right away. When I was 18, I worked in a steel plant in Detroit, and one of the jobs I had to do was go in and repair the roof on this anhydrous ammonia plant. And the first day I got there, I said, "Dear Lord in heaven, help me through this day," because I couldn't believe how bad it smelled. It was like I was getting constantly hit in the nose with a sledgehammer at this place. And these guys were just walking around like nothing was, just whistling Dixie. And then I was there for a week, and by the end of the week, I was just walking along whistling Dixie. I couldn't smell it any longer. My smell had fully adapted to that smell. So hair movement. You can run your hands through your hair. You feel that feeling of running your hands through your hair. The hair has changed its position. But once it's changed that position, those receptors that are around the root hair plexus, they adapt quickly. And you don't feel the position of every hair on your head any longer. You touch one of them, you can feel it change. But once it's stopped changing, it adapts once again. Cutaneous pressure, sitting on a table. You feel yourself sit, but then you quickly adapt to pressure on the skin. All right. So if we look here, these are tonic receptors, and this is stimulus. We're going to take receptor. We're going to add stimulus to it. We have the spike of the potential at that sensory receptor, and there's a little bit of adaptation, but it's still depolarized, all right, until the stimulus is removed. Here, phasic receptors spike on when there's a change in the stimulus, but the stimulus is constant here, and it rapidly adapts until we remove the stimulus, and then there's this brief off response that also rapidly adapts. Guys, follow? You're thinking deeply? Yes.
So is this over the time of one action potential, or is this over the time of the stimulation of the receptor, which it could be happening for multiple action potentials in a row?
Yeah, this would be multiple action potentials, yeah. All right, so here is another one of my vast outlines of the topics we're going to cover for you guys to give you a way to look ahead. We're going to talk about some of these fiber types. We're going to go through the types of senses in the body. Eventually, I will talk about the special senses, particularly sight and hearing. Each will get their own lectures. And then I'm going to go through these functional categories of receptors. Nociceptors, pain, thermoreceptors, receptors to heat, chemoreceptors, mechanoreceptors, pressure receptors, and then the proprioceptors. This is just an outline that you can use for your reference. All right, with the last little bit here, I want to try to stumble our way through some of the types of general or some aesthetic senses. There are three classes of mechanoreceptors. First, the tactile receptors, and I'm going to go through these in terms of their sensitivity, all right, and their complexity of design. So the first are just these free nerve endings. These are highly tonic receptors with very small receptive fields. This is giving us pain or nociception and temperature in the skin and mucosa. Look, very easy to set these off. Very easy to set these fibers off. Not much is modulating or attenuating the stimulus that would set these fibers off. Tonic receptors, a pain receptor. You don't really want a pain receptor turning off, adapting to it, because why would that be?
Because then you'd get hurt.
Then you'd get hurt. All right, root hair plexus. Also very unmodified nerve ending. However, these adapt very rapidly. They're wrapping around the base of a hair, and this is the example I told you. When you move a hair, it's telling you information about the first derivative of the stimulus, the change in the stimulus. All right? So this is good at detecting initial contact and any subsequent movement. Something's crawling through your hair, good to know. If it's just standing up there, then it's probably a barrette or something. All right. Next are these Merkel cells, tactile discs. So these tactile discs begin to be a little bit modified. These tactile discs are associated with these specialized Merkel cells which attenuate, modify slightly the kind of stimulus, the kind of tactile stimulus that's going to be responsible for setting these fibers off. So fine touch and pressure. These are tonic receptors, and they have very small receptive fields, very small receptive fields. Merkel cells are the kind of cells that you enjoy getting stimulated. Then there's the Meisner corpuscle. A Meisner corpuscle has the nerve ending surrounded in this collagenous capsule or corpuscle. Meisner, named after the histologist that first identified it. This will be my last slide. So this is sensations of fine touch, pressure, also low-frequency vibration. Low-frequency vibration. The adaptation happens after approximately a second of contact. These are pretty large structures. They're the favorite of histologists on histology exams because they're easy to identify underneath a slide. All right, they're sticking up in the dermal papillas, in the papillary layer of the dermis, right below the epidermis. So here's one. These are the ones that are particularly fun for humans to have stimulated. They're ones in the fingertips, the eyelids, the lips, the nipples, the genitalia. All right, all those places that humans like to be touched. All right, so I'll leave it there. We'll come back to lamellated corpuscle. I don't want to go over. And I'll just finish up the last few of these slides, then we'll start moving into central nervous system next lecture. Okay, questions? Yeah, there'll be a quiz at the beginning of next class over, what was that, muscles and cell physiology, the one lecture of cell physiology and like membrane potential stuff, and then into what I talked about on muscles. So next week, or on Wednesday. Any other questions?